Welcome to today's lecture on the fascinating world of cell biology and the various cutting-edge techniques used in medical research. In this lecture, we will dive deep into the realm of molecular biology and explore the intricate details of several techniques that have revolutionized our understanding of genes, genomes, and cellular processes. From real-time PCR to CRISPR-Cas systems, we will unravel the complexities of these techniques and their applications in genome analysis and manipulation. Real-time PCR, or qPCR, is a technique that amplifies and quantifies specific DNA sequences in real time. It uses fluorescent dyes or probes to monitor the amplification process and provides quantitative data on the initial amount of DNA. This powerful tool has applications in gene expression analysis, pathogen detection, and viral load quantification. In the mutation detection by PCR and DNA sequencing section of the video, we explain how PCR and DNA sequencing are used to detect mutations in genes or genomic regions. We cover the steps of PCR amplification and DNA sequencing using Sanger sequencing or next-generation sequencing methods. The applications of mutation detection in various fields are also discussed. RNA-seq is a powerful technique used to study gene expression. It can detect both known and new RNA molecules in a sample. This method involves isolating RNA, converting it into cDNA, preparing a library, and sequencing. The resulting data can be analyzed to understand gene expression, alternative splicing, and identify new transcripts. RNA-seq has revolutionized our understanding of biology and disease. Next-generation sequencing technologies have transformed genomics by allowing rapid, cost-effective sequencing of entire genomes. Whole genome sequencing, WGS, involves DNA extraction, library preparation, sequencing, and bioinformatics analysis. Variations in the sequence data help reveal disease-causing genes and advance personalized medicine and evolutionary biology. Metagenomics is a powerful approach used to study microbial communities without the need for culturing individual organisms. This technique involves the direct extraction of DNA from environmental samples, amplification using PCR, and subsequent DNA sequencing. Metagenomic analysis provides insights into the diversity and functional potential of microbial communities. By identifying and comparing the genetic material present in a sample, researchers can understand community dynamics, investigate interactions, and discover novel microbial species. PCR amplification plays a crucial role in metagenomics, as it enables the targeted amplification of specific genes or regions of interest. DNA sequencing technologies, such as NGS, provide high-throughput sequencing of the amplified DNA fragments, facilitating comprehensive analysis of complex microbial ecosystems. ChIP-seq is a technique that helps us understand how proteins interact with DNA. It involves fixing proteins to DNA, breaking it into smaller fragments, and then isolating the protein-DNA complexes. Sequencing the DNA can reveal the binding sites of certain proteins, giving us insight into gene regulation and chromatin structure. This knowledge is important in studying development, disease, and epigenetics. DNA barcoding is a technique used for species identification based on short and highly conserved DNA regions, known as DNA barcodes. These DNA barcodes are unique to each species, allowing for rapid and accurate species identification, even in cases where traditional morphological methods fail. The DNA barcoding process involves the amplification of the target DNA barcode region using PCR, followed by DNA sequencing. The obtained sequences are then compared to reference databases to identify the species. DNA barcoding has revolutionized biodiversity research, forensic sciences, and conservation biology. It enables rapid and accurate identification of species, facilitates the discovery of new species, and aids in assessing the impact of environmental changes on biodiversity. Genotyping, the identification of genetic variations within an individual or population, is essential for understanding genetic predisposition to diseases, population genetics, and personalized medicine. High-throughput genotyping utilizing PCR-based assays has significantly accelerated the genotyping process. PCR-based genotyping techniques, such as allele-specific PCR, multiplex PCR, and real-time PCR, allow for the simultaneous amplification and detection of multiple genetic markers. These markers can be single nucleotide polymorphisms, SNPs, insertions, deletions, or copy number variations. High-throughput genotyping plays a crucial role in genome-wide association studies, GWAS, pharmacogenomics, and forensic genetics. By examining large numbers of individuals and genetic markers, researchers can unravel the genetic basis of complex diseases and design personalized treatment strategies. Epigenetic modifications are chemical changes to DNA and histone proteins that regulate gene expression. PCR and DNA sequencing help study epigenetic modifications. Understanding them is vital in unraveling gene regulation mechanisms and their involvement in development, disease, and potential therapeutic interventions. Comparative genomics involves the analysis of DNA sequences from different organisms to understand evolutionary relationships, identify conserved elements, and uncover species-specific traits. DNA sequencing plays a central role in comparative genomics, providing vast amounts of data for analysis and interpretation. DNA sequences from multiple organisms are aligned and compared using computational tools to identify similarities, differences, and conserved regions. Evolutionarily conserved regions often indicate functional significance, such as protein-coding genes or regulatory elements. Comparative genomics has shed light on diverse aspects of biology, including evolutionary history, adaptive changes, and genome evolution. It has enabled the understanding of human biology through comparisons with model organisms and facilitated the discovery of new genes and functional elements. By harnessing the power of PCR and DNA sequencing, single-cell genomics allows researchers to delve into the intricacies of individual cells. This technique uncovers hidden cell types and heterogeneity overlooked in bulk studies. Amplification of DNA from single cells enables library preparation and sequencing, leading to breakthroughs in areas like developmental biology, stem cell research, and cancer biology. In summary, CRISPR-Cas systems are a powerful tool for editing DNA sequences. They consist of RNA-guided nucleases, such as Cas9, and guide RNAs that direct the nuclease to specific targets. This system allows for precise modifications to DNA, with potential applications in gene therapy and genetic engineering. In conclusion, PCR and DNA sequencing techniques have revolutionized cell and molecular biology. These tools enable us to study various aspects of genetics, such as gene expression, mutations, and epigenetic modifications, leading to advancements in medical research and personalized medicine. This field holds immense potential for future discoveries and improvements in healthcare.